In this video, I will be reviewing the Infectious Disease Society of America IDSA guidelines for the treatment of COVID-19. All of us want to help our patients with COVID-19, and we have heard about or read papers touting different treatments. As an associate editor of the Journal of Infectious Diseases, I can personally attest to the importance of rigorous peer and editorial review. There are now 1,558 COVID-19 articles available on the internet through medrxiv.org. Too many of these papers have circumvented the usual research steps, including failure to secure IRB approval, and have included the same patients in several studies, a process discouraged by nearly all peer-reviewed journals. Secondly, they have not been subjected to vigorous peer review or have not undergone any peer review before being released on the internet. When the due diligence from editors and reviewers is sidestepped, unnoticed errors in data and calculations, incomplete reporting of methods and results, as well as underestimation of study limitations are likely. Third, these papers often contain publication bias. In the interest of showing promising data in the race to achieve recognition, there may be added inclination to publish positive results and disregard negative ones. The exact extent and impact of these considerations are currently uncertain, but were acknowledged in the development of the IDSA guidelines. To address these concerns, the Guidelines Committee used an extremely helpful tool that is now recommended for all clinical studies, GRADE, G-R-A-D-E, which stands for Grading of Recommendations, Assessment, Development, and Evaluation. A mouthful, but critical for deciding what treatments will truly be of value to our COVID-19 patients. You may ask, how should we decide if a treatment is of value? I recommend asking this question. Will the therapy in question improve the health and well-being of my patient? If the answer is yes, this therapy should be given. If the answer is no or I don't know, in most cases the therapy should not be administered. When the answer is I don't know, the treatment should only be ordered if the patient is placed in a controlled clinical trial and a specific consent form approved by the IRB has been signed by the patient following a clear explanation of the risks and benefits of that treatment. Let's look at the IDSA Guideline Committee's assessment of the evidence. This multidisciplinary team of experts included pharmacists, research methodologists, and infectious disease experts from multiple academic institutions and health systems that are listed here. To show how this expert panel applied GRADE, let's look at one French study on the efficacy of hydroxychloroquine and hydroxychloroquine plus azithromycin for reducing viral count as assessed by nasopharyngeal RT-PCR for COVID-19 patients. This study consisted of 36 COVID-19 patients. 20 received hydroxychloroquine and 16 served as control patients. For each group, RT-PCR positivity was determined at day 6 post-inclusion in the study, and the percent positive patients compared. The mean age of the controls was 37 years, and the treatment group 51 years. The control group had a higher percentage of asymptomatic and upper respiratory infections as compared to the treatment group, 87 versus 70%. Why are these demographic differences important? Because PCR test positivity is known to be higher in younger individuals and in those who have upper respiratory symptoms or are asymptomatic. Furthermore, the control group either came from a different hospital than those receiving treatment or had refused to take hydroxychloroquine. My conclusion as an editor, the increased percentage of negative PCR tests in the treatment groups can be explained by demographic and clinical differences in the two populations being studied. In my view, This paper should not have been accepted for publication because it was seriously flawed. Now let's look at the IDSA analysis using grade. Look at their assessment in the bottom column. The study could not be considered randomized or controlled, but rather was a poorly designed observational study. Observational studies have repeatedly been found to draw spurious and incorrect conclusions. They felt the risk for bias for this study was very serious. Inconsistencies and indirectness in design of the study were also rated as seriously flawed. They also felt imprecision of the analysis was serious, yielding the conclusion that the certainty was very low that hydrochloroquine treatment reduced the risk of viral failure. Now let's look at the IDSA grade analysis for other treatments of COVID-19. Lopinavir/ritonavir, certainty of benefit or non-benefit was again assessed as very low because of bias and imprecision. IL-6 inhibition by tocilizumab treatment versus no treatment in patients with severe COVID-19 pneumonia. Again, the certainty of benefit was assessed to be very low because the studies were observational, risk of bias was serious, and imprecision of analysis was serious. For treatment with intravenous administration of convalescent plasma versus no convalescent plasma, the certainty of benefit was rated as very low. What about remdesivir? A recent case series of 53 patients with severe COVID-19 pneumonia who had received remdesivir under compassionate use protocol were reported to clinically improve in 68% of cases with follow-up at 18 days, with a 13% mortality and a generally acceptable toxicity profile. This paper was recently published in the New England Journal. However, the IDSA committee noted there had been no comparison group of similar patients who received standard care at the participating institutions. The IDSA is awaiting the randomized clinical control trials before making any recommendations about remdesivir. The overall conclusions by the IDSA expert panel: none of these treatments should be administered unless the patient is part of a randomized clinical controlled trial.